My name is Vaibhav. I'm a PhD student at UC Riverside. Have you ever noticed that when your fruits get really ripe, tiny little flies start invading your kitchen? Well, I study how fruit flies taste, and what I've realized through careful experiments and really fancy science is that they taste really bad. Boo! The brain and the taste organs in a tiny little fruit fly allow it to eat lots of tasty food. But the fly also needs this food to gain energy and to survive. And you might be wondering, why are you studying how a fruit fly is able to taste food? Why is that important? Well, you and I are actually pretty similar to insects in the way that we taste. We both use cells called taste receptors in order to figure out the taste of what we eat. Today, we're going to do an experiment to see whether the fly prefers one of two different kinds of chemicals, sucrose or sodium chloride. How would you describe the taste of sucrose? If you said sweet, well, that's exactly right, because sucrose is a sugar that we often find in fruits. What about sodium chloride? Well, sodium chloride is the chemical name for salt, which we use every day in our cooking, therefore has a salty taste. So we're going to let the fly choose which kind of food it prefers to eat. Pure sucrose or pure salt. When we raise flies in the lab, we typically store them in big bottles containing our homemade fly food recipe so that they have plenty of space to move around and they never run out of food. Here you can see a bunch of flies moving around inside these bottles. Flies lay their eggs in the food at the bottom of the bottle, which eventually develop into larvae. Within a short time, larvae, which look like small white worms, start climbing up these bottles and become what are called pupa. Pupa undergo a process called metamorphosis, and at the end of that process, they emerge from their pupal shell as an adult fly with wings. To set up our experiment, the first step is to collect 10 male flies and 10 female flies from bottles like the one I showed you earlier. Then we transfer them into vials like these. We'll let them hang around here for a few days so that they can mature properly and get used to this environment before actually starting the experiment.
Several days later.
Before we let our flies choose between sugar or salt, we first need to starve them for 24 hours. We do this so that our insects will be hungry by the time that they are exposed to food. To starve the flies, we roll up 2 pieces of fancy lab-grade tissue paper into a tiny, tightly packed ball and just toss it into an empty vial. Or if you have bad aim like me, you just stuff it in. Then we use a pipette to collect some water and wet the paper towels in the vial so that the flies can use their proboscis, which is kind of like a tongue, to draw up water. But other than that, they have no access to food until the experiment starts.
24 hours later.
Today is the big day. After a few days of preparing and setting up, we are finally doing our experiment. The first step is making sure we have a chamber for our insects to walk around in, so that they have the ability to sample the sucrose or salt that we're going to deposit onto this plate, a small petri dish. We normally think of sugar and salt as solids, but in order for the fly to ingest food, we need to provide it with a softer, semi-solid form of our foods. To do this, we are going to create liquid solutions of sucrose and salt by dissolving them in water and making them the same concentration. We're then going to add a red dye to the sucrose and a blue dye to the salt so that we don't mix the foods up. We then add a tasteless substance called agarose to each solution. Agarose is a substance that behaves like jello when you heat it up. This will ensure that our food is not too watery so that it doesn't spill everywhere when we add it to the plate. We just give everything a good mix, and now we can place the food on our plate. We're using another kind of pipette to transfer the food from the small tube onto the plate in a series of droplets. We use pipettes for two reasons. First, we don't want to contaminate anything with our hands, and second, we can control exactly how much liquid we dispense. We want to be consistent and make sure we're dispensing an equal amount of both taste chemicals so that we do not bias the flies into choosing one food over the other. Remember, the blue liquid is our sodium chloride and the red liquid is our sucrose. At this point, we're ready to add our flies to this plate. These flies have been starving for 24 hours and are hungry for some food. To do this, we expose them to carbon dioxide, which anesthetizes them or makes them pass out. If you look closely, you might notice that they are dropping like flies, which makes sense because they are flies. And now that they've been knocked out, time is running. We have to quickly tap the vial to push the flies out onto the cover of the plate. Once all of them are out, we make sure the lid is on tight and place them in this super high-tech, state-of-the-art, humidity-controlled chamber, also known as a wet styrofoam box. Now we let them feast for 2 hours.
2 hours later.
So I've let the flies feed for 2 hours, and then I immediately place the Petri plate into a freezer that is minus 80 degrees Celsius in order to prepare the flies for dissection. During this process, I want to open up the gut of each fly to see what color it is. If it's red, I know they ate sucrose. If it's blue, I know they ate salt. If it's purple, I know they ate both sucrose and salt. And if it's clear, I know they didn't eat anything at all. Because flies are so small, I'm going to dissect them under the microscope using very sharp but very small tools called forceps. Here's an example of what a fly might look like under the microscope. It's pretty obvious that this fly chose the sucrose, which is our red food, but I'm going to open it up just to double check. Yep, still red. This guy, on the other hand, wasn't very hungry. His gut is still clear. With the power of fast forward, I'm going to zoom past all the dissections and do some quick analysis to let you know the answer to the question we've all been waiting for. On a scale of 0 to 10, where 0 means that flies prefer sodium chloride, 5 means they prefer both sodium chloride and sucrose equally, and 10 means that flies prefer sucrose, the flies scored a 9 out of 10. This means that when offered the same amount of sucrose and sodium chloride, the flies really preferred sucrose. This makes sense when we think about it. Think about how good a tablespoon of raw sugar tastes compared to a tablespoon of pure salt. Flies, like us, often associate things that taste good as safe, viable sources of food, and tend to stay away from things that taste bad, fearing that they may be toxic. We can use this same kind of experiment to compare many different kinds of foods with each other. We can compare different concentrations of the same food. We can compare foods that taste similar but have different nutritional values, such as table sugar versus low-calorie sweeteners. And we can also learn a lot about how the neurons that sense this taste communicate information to the brain so that the animal can make a decision about whether to continue or to stop feeding. I hope you enjoyed this journey and have a happy Brain Awareness Day.